The overall goal of this procedure is to process a human vestibular schwannoma sample received from the operating room for primary cell culture and collection of tumor secretions.
This method can help answer key questions in the field of vestibular schwannoma research, such as how best to receive, store, and process a primary human tumor sample intended for use in multiple biomolecular assays.
The main advantage of this technique is that it makes use of a minimally manipulative culture method to propagate primary human vestibular schwannoma cells.
The implications of this technique extend toward therapy for vestibular schwannoma because promising therapeutic candidates can be tested directly on cultured cells.
Begin by transferring the specimen container holding the freshly harvested vestibular schwannoma or great auricular nerve sample from the ice used for transportation from the operating room to a laminar flow hood in the laboratory. Use sterile forceps to carefully transfer the specimen to the first of 3 1.5 milliliter tubes filled with 1.4 milliliters of 1x sterile PBS. Close each tube once it contains the specimen and shake gently to wash. Place the sample in a dry 60 millimeter petri dish and use forceps to remove all dead tissue in areas with prominent blood vessels. Use forceps or a scalpel blade to divide the sample into separate pieces for RNA preservation, protein extraction, secretion collection, fixation, and cell culture.